Adenylyl cyclase is a membrane-bound enzyme whose catalytic domain is activated by the GTP-bound form of the stimulatory G-protein alpha subunit, or G-alpha-S for short. Activated adenylyl cyclase converts ATP to cyclic AMP, which then acts as a second messenger that relays the signal from the G-protein coupled receptor to other components in the cell. In most animal cells, cyclic AMP activates cyclic AMP-dependent protein kinase, or pKa. In the inactive state, pKa consists of a complex of 2 catalytic subunits and 2 regulatory subunits. The binding of cyclic AMP to the regulatory subunits alters their conformation and liberates the catalytic subunits, which are now active and phosphorylate specific target proteins. In some endocrine cells, for example, the activated pKa catalytic subunits enter the nucleus, where they phosphorylate a transcription factor called CREB. Phosphorylated CREB then recruits a CREB binding protein. This complex activates transcription after binding to specific regulatory regions that are present in the promoters of appropriate target genes.
Okay. So what we have discussed in this animation, through this animation, is the G-protein coupled receptor. What I have shown you before. Okay. So this G-protein coupled receptor, there are different receptor cells. So let me come here. Yeah. Let me change the color. So here, in this case, we have a G-protein coupled receptor here. That receptor is made up with 7 transmembrane receptors. Okay. Normal examples of this kind of signaling is a signal molecule come and sit into this this receptor, and finally then after that the signaling pathway goes on. It activates the adenylyl cyclase, and via there's protein kinase A, and this kinase in phosphorylate, as we have seen, and it will induce the CREB, and finally it will induce the gene. It will trigger the gene regulation, and then the cell proliferation. All these things will go on. Okay. And what are the chemicals that act as signal molecules in this G-protein coupled receptor pathway are chemokines, different types of hormones, and neurotransmitters can also be a part of this signaling molecule. Okay. So this is the first pathway we have discussed. Now we'll discuss about the Pi3 kinase pathway. So in this Pi3 kinase pathway, what will happen? Yeah. So this Pi3 kinase pathway, what will happen here is the receptor tyrosine kinase, or the tyrosine kinase receptor. In this tyrosine kinase receptor, this is not made up with 7 transmembrane receptor. The tyrosine kinase receptor is made up with the transmembrane region, and it has a dimer. So we have a similar 2 subunits. The subunits are not attached with each other in primary space, but whenever the signaling molecule attaches with the extracellular space of the signaling place, like that, then it will trigger the autophosphorylation of those tyrosine kinase molecules. So one phosphate is attached to one subunit, and that this phosphorylation triggers the attachment of another phosphate to another subunit, and as a result of this autophosphorylation, those structures are dimerized, and then it will change a certain kind of cell signaling via the activation of Pi3 kinase. And the pathway is naming after this Pi3 kinase that suggests that the phosphatidyl inositol 3 kinase pathway. Okay, and that will trigger the AKT, which is another protein, and finally it leads to the formation of different pathway. It will trigger it throughout this place, and not only this pathway is important for the gene regulation purpose, but also this pathway regulates the apoptosis signaling in this place, as we know, because the product of this AKT, or this pathway, that AKT that can inhibit this bad protein, and this bad protein can trigger the apoptosis, and this AKT molecule will block the activity of bad protein, and as a result of it the apoptosis will stall, or the programmed cell death will stall. So any problem in this AKT can convert a cell to not die as a result of the cell signaling of natural programmed cell death process, or in turn we can say the mutation in this molecule, or the change in or malfunctioning in this region of cell signaling, can in turn make a cell cancerous. Okay, so now let's see what are the importance of this pathway and how this pathway is go on. So take the arrow. Yeah.
The Pi3K Act signaling pathway. The Pi3K Act signaling pathway consists of many activators, inhibitors, effectors, and second messengers. Because of its many loops and branches, the pathway is very complex and far from understood. Here, only the most important circuits will be presented in a simplified way. Activation of Pi3 kinase. The Pi3K Act pathway starts with the activation of phosphatidylinositol 3 or Pi3 kinase. This activation can be accomplished via 3 different pathways. 2 pathways begin with the activation of a receptor belonging to the family of receptor tyrosine kinases by an extracellular growth factor. Binding of the factor leads to the dimerization of the receptor monomers and the heterologous autophosphorylation of the monomers. Depending on the receptor, different proteins may bind to a phosphorylated domain. The insulin receptor substrate 1 or IRS1 binds to the activated IGF-1 receptor. For simplification, this receptor is depicted as a dimer. Receptor-bound IRS1 serves as a binding and activation site for the Pi3 kinase. In addition, Pi3 kinase may bind directly to a phosphorylated receptor tyrosine kinase. A completely different mechanism of Pi3 kinase activation begins with the small membrane-bound GTPase RAS. By binding to active GTP-bound RAS, Pi3 kinase is activated. Formation of the second messenger phosphatidylinositol 3,4,5 trisphosphate PIP3 and activation of ACT. At the second level of the pathway, the second messenger phosphatidylinositol 3,4,5 trisphosphate or PIP3 is formed. This leads to the activation of the serine threonine kinase ACT. The active Pi3 kinase migrates to the inner side of the cell membrane and binds to phosphatidylinositol 4,5 bisphosphate or PIP2, which is a regular component of the membrane and is anchored by its 2 fatty acids in the lipid layer of the membrane. Pi3 kinase is able to phosphorylate PIP2 to PIP3. PIP3 can activate the kinase ACT, which is also called protein kinase B and was named after its homologous protein in the retrovirus ACT8. Downstream effects of ACT. ACT, a serine threonine kinase activated by PIP3, is a proto-oncoprotein with many substrates and effects. Here only 3 effects will be exemplified. The best known effect is the inhibition of apoptosis, programmed cell death. ACT binds to Bax and hinders its ability to form holes in the outer mitochondrial membrane. In the absence of ACT, these holes lead to apoptosis via the caspase cascade. Another important effect of ACT is the activation of protein synthesis or translation. This effect is performed by a multi-step protein cascade. The cascade begins with the activation of the protein Reb, which activates mTOR. mTOR itself interacts with and activates the translation factor S6K. By binding to the large subunit of ribosomes, S6K activates the translation of mRNA into protein. In addition, ACT may lower the concentration of the protein FOXO by phosphorylating FOXO. Phosphorylated FOXO is a substrate of the enzyme ubiquitin ligase, which transfers ubiquitin peptides onto the protein. These peptides are symbolized by small black dots. Subsequently, ubiquitinated FOXO is destroyed by a complex of proteases, the so-called proteasome. In this way, ACT prevents the tumor suppressor protein FOXO from inhibiting proliferation.
Okay, So what you have seen in this picture, or in this, not picture, an animation, the control of the Pi3 kinase pathway, as long as the importance of the Pi3 kinase pathway establishing cancer, and also how Pi3 kinase pathway is making a cell into a cancerous cell, and how that can be prevented. And we can know how that can be prevented because we know the pathway. So this AKT, or serine threonine kinase, is really very important molecule in this Pi3 kinase system. As we can see that it will trigger to make this cell to make a response that can stop that cannot stop the apoptosis, that can stop the apoptosis mechanism or programmed cell death mechanism. As a result of it, the cell will go on and on, it will never die. And not only that, but also the FOXO gene, which will produce some proteins. Those proteins act as, normally they can act as the tumor suppressor proteins, but due to the result of the ubiquitination of those FOXO by this AKT molecule, can leads to the massive proliferation of the cell. Okay, so this Pi3 kinase pathway is interlinked with this apoptosis pathway, and also this is interlinking or cross-talking between the different pathways that finally makes a cell to function like a way that we understand nowadays, to function like a really complex material, a really complex system rather. Okay, so we have talked about the G protein coupled receptor. We can also we have also talked about Pi3 kinase, or most importantly the AKT kinase pathway. And in this case, growth factors are the chemical signal, or growth factor acts as chemical signals in this Pi3 kinase pathway, because growth factors can bind, and they finally leads to the maturation of the cell and the cell proliferation. And if a cell had to proliferate more of the time, or the most of the time, then the cell can also have to trigger this apoptosis. Okay, so a cell, when it thinks about the massive proliferation or proliferating beyond its normal capability, then it must stop the apoptosis pathway. And not only must stop the apoptosis pathway, and it also go against some proteins, some natural proteins produced by the cells, like FOXO, which are the tumor repressors. Okay, so that's a very important consideration. And we can also see this linkage between this G protein coupled pathway and also the Pi3 kinase pathway. That linkage is very important in this case too. So they are not a single pathway is going on its own. So they are there are different cross talking. There are different cross passes of different signaling from one pathway to another pathway. You can see here. So that's a very fascinating part. Okay. So now let's talk about the MAP kinase pathway, or we can say the RAS pathway or MAP kinase pathway, and what is the role of this MAP kinase pathway, and how that pathway is accomplished. Again, in this MAP kinase pathway, we will see the receptor tyrosine kinase as a receptor, and we can also see a RAS protein, which is a G protein bounded with the cell membrane, and the act of this RAS protein. Because we have seen this RAS protein in Pi3 kinase pathway, but not in greater detail. But we will see this in a greater detail in this case. Okay. So now let me take the arrow.
The MAP kinase signaling pathway. The MAP kinase signaling pathway in the normal cell. The designation MAP kinase, abbreviated MAPK, originates from one of the first members of this protein family, the Mitogen-Activated protein kinase. The MAP kinase pathway begins with the binding of a ligand, for example EGF, to the extracellular portion of a membrane-bound receptor of the family of receptor tyrosine kinases, abbreviated RTKs. Ligand binding leads to the dimerization of two subunits of the receptor tyrosine kinase. At the inner side of the receptor, tyrosine kinase domains catalyze phosphorylation of itself and also of the other subunit. Next, the growth factor receptor-bound protein 2, GRAP2 for short, can bind to the phosphorylated RTK. The protein SOS, named after its homologue son of sevenless in Drosophila, is able to bind to the membrane-bound protein RAS. Inactive RAS is bound to the nucleotide guanosine diphosphate, GDP. SOS catalyzes the exchange of the RAS-bound GDP against GTP, guanosine triphosphate. This exchange leads to the activation of the RAS protein. In its active GTP-bound state, RAS is able to bind to several effector proteins. The kinase BRAF is one of the most important effectors of RAS. Active BRAF phosphorylates and activates the kinases MEK1 and 2, which in turn phosphorylate and activate the kinases ERK1 and 2. Finally, the kinase Cascade leads to the activation of transcription factors of the AP1 family. The abbreviation AP1 stands for Activator Protein 1. The best-known AP1 proteins are the transcription factors Jun and Phos. After their activation, Jun and Phos move to the cell nucleus, where they form a heterodimer and bind to an AP1 motif of the DNA. This leads to the expression of many genes encoding, for example, growth factors, cyclins, and cytokines. As a direct consequence, cell proliferation is activated. In the normal cell, the active RAS-GTP complex is inactivated shortly after its activation. In this way, a permanently active MAP kinase signaling pathway and undesirable effects are avoided. RAS-GTP is inactivated by the GTPase activating protein, also known as GAP. GAP binds to RAS-GTP and increases the very weak GTPase activity of RAS by several orders of magnitude. GAP achieves this activation by providing a domain that assists the RAS protein in the hydrolysis of GTP. The bound GTP is hydrolyzed to GDP. GDP-bound RAS is no longer active and can no longer bind to BRAF. As a result, the MAPK signaling pathway is turned off. The MAPK signaling pathway in a tumor cell with RAS mutation. Mutation of the RAS gene may have dramatic consequences for the cell. Mutated RAS protein is activated just like the normal protein. Mutated active RAS-GTP is also able to bind and to activate the kinase BRAF. A major difference between the mutated and the normal RAS-GTP protein is that the mutated protein loses its capability to be inactivated by the GTPase activating protein GAP. GAP can still bind to mutated RAS in its GTP-bound form. However, GAP is not able to provide the domain that is crucial for the activation of the RAS-GTPase. GTP is not hydrolyzed to GDP. Consequently, mutated RAS-GTP stays active. The kinase BRAF stays bound to RAS-GTP and remains active. BRAF phosphorylates and activates an increasing number of MEK1 and 2 proteins. In turn, MEK1 and 2 activate ERK1 and 2, which activate Jun and Phos. Thus, the kinase cascade is not turned off. A permanently activated MAP kinase signaling pathway results in continuously activated proliferation, which is an important feature of a tumor cell.
Okay, now let's focus on what we have studied in this pathway. So in this pathway, which is called the MAP kinase or Mitogen-Activated Protein Kinase Pathway, there are lots of different diverging ways like MAP kinase, MAP kinase, MAP kinase, and all these different parts, and also the ARC, which will activate Phos and Jun, as we have seen. And in this pathway, the gene regulation is done in such a way that the proliferation of cell is taken place. Okay, as we know, if the proliferation of cell is taken place over the normal system or normal proliferation, that can cause cancer or can cause the uncontrollable cell growth or which is called the cancer. Okay, in this whole pathway, the receptor molecules or the signaling molecules are most of the time the epidermal growth factors or EGF, and also also different growth factors are responsible for these purposes. Okay, so in this particular system, or in this particular pathway, RAS molecule is really important, because as you've seen, this RAS molecule, you can see here, are linked with different pathways, from pathway of G protein coupled receptor as well as the pathway of Pi3 kinase pathway. The RAS protein is a cross-linking molecule of several pathways. So any problem in this RAS, for example any mutation which causes the structural alteration of the RAS, can eventually cause diseases like cancer, as we have seen in the animation. Okay, so if the RAS molecule is activated, or it is turned on for many times, or for many minutes, or for several sets of times, then this RAS molecule can create or can produce the cancerous cells, or can produce the property of cancer. That means the massive proliferation and blockage of apoptosis. Okay, so that's why this pathway is important. So why this, we can see this cell signaling pathways really have to have a tight control over what they are doing. So if any single part of them, any single cascade of this pathway, any single molecule of this pathway is mutated, or it is blocked, or it is restricted from its activity, then they leads to formation of massive bad effects in the cellular applications. Okay, now let's talk about the JAK-STAT pathway in this case, or the pathway where we have normal cytokine receptor, and cytokines only act as the chemical signaling for the JAK-STAT pathway. Why we talk about the JAK-STAT pathway? Again, this JAK-STAT pathway will end up to the activation of caspase 9, which is a really important molecule for apoptosis purposes or programmed cell death purposes. Now one question can be arised, that why apoptosis is required, why we need to kill a cell after some time? Because a cell have its lifetime, and in those lifetime of the cell, a particular limit of time of the cell works properly. After that, the cell can produce some very dangerous materials which can be toxic for other cells, or the cell will lost its functioning. So we need to feed that cell much, and the application or the work what we can have from the cell is getting slower. That's why I need to kill those cells routinely. So we need to clean up, just like we need to clean up our house. So we have a house, we have different things scattered in our house. The bad or junk materials have to be discarded, and the good materials has to be placed in a different mannered way. That's how the cell works together, because we have trillions of cells. So if we kill those bad cells, that's better for us. So the killing of all those cells never turns on the losing of cells. We have other cells, and we also have the cellular multiplication or cellular duplication, through which we can achieve the number of the cell, or make the number of the cell constant. Okay, but after the certain age in humans, after a certain age, when the cell can have the apoptosis pathway, or cell will die via the apoptosis pathway, and no longer new cells are produced. In those situations is called the aging. Okay, so in those aging situations, we do not have anything to do, because it's a rule of nature. Recent work is going on to prevent aging, but still aging is a natural process. In those natural process, the cells lose its function, they have to be killed, and new cells, or the propagation of new cell, or the production or productivity of the new cell is getting slower. That's what is happening. So now let's focus on the JAK-STAT pathway, and what is the implication of the JAK-STAT pathway in biological systems, in cellular systems. Okay, so this pathway is not having the narration, so I have to narrate this pathway. So let me take, okay, so here is the cytokine signaling. So we have cytokine receptors. These two are the cytokine receptors, having two different domains, which have the intracellular domain or JAK domain, and also the extracellular domain. As we can see in this intracellular domain, there is a tyrosine residue. So whenever cytokine sits in, the tyrosine residues are there. These tyrosine residues are phosphorylated. So whenever the receptor binds, tyrosine residues are phosphorylated, and as long as tyrosine are phosphorylated, the STAT proteins are there. Those STAT proteins comes together, and they are also phosphorylated by the tyrosine present there. After the phosphorylation of those tyrosine containing STAT molecules, the STAT molecules are dimerized with each other, as you can see here. So what happens, these JAK molecules are there, JAK receptor is there. After the autophosphorylation, they will phosphorylate the STAT molecules, and now the STAT molecules make a dimer to attach with themselves, and after making the dimer, it will enter into the nucleus. It will bind with the STAT binding sequence, which is present in a certain region of our DNA. After the binding with the STAT binding sequence, it will trigger transcription or transcription factors or activated proteins. After the production of all these transcription factors, the gene transcription or the gene for the cytokine response is activated, and whole system is going on throughout this pathway. Okay, so what we have seen in this case, this JAK molecule is activated, and it further autophosphorylates and phosphorystats, and finally it leads to the formation of those transcription factors or proteins. In those cases, the pathway or the animation is really small, but ultimately what we have seen in animation, it leads to the production of few proteins, and caspase 9 is one of those proteins. What caspase 9 do in most of the cases, caspase 9 or caspase 8? Caspase 8 is produced via the FASR pathway. In this case, we can see here. We will not going to discuss about FASR in detail because they are more or less similar pathways here. So the production of this caspase 8 or caspase 9, they are the death proteins. What we mean by death protein is those proteins have to do something with the death of the cell, that means the apoptosis of the cell. So caspase 9 and caspase 8, they will make a whole, they can make DNase enzymes, they can induce DNase enzymes, and what those DNase enzymes will do, they will chew up the genes or chew up the DNase which present inside the nucleus. Once the production of caspase 8 or caspase 9 is done in huge amount, then they produces those DNase, and DNA will chew up all these things, and finally when the nucleus or genetic material is degraded, the whole cell is going towards the apoptosis pathway. Okay, and that's what is happening throughout this JAK-STAT pathway, what we have just studied. Okay, so in all those different pathways, in G protein coupled pathway, in Pi3 kinase pathway, as well as the MAP kinase pathway, what we have discussed, we are going towards a particular point, a point of programmed cell death or point of apoptosis. So now let's talk about this apoptosis or programmed cell death pathway in really more great detail. But before going to that, I must talk about the control of the cell signaling, control of the signaling via the G protein coupled receptor, and as well as the control of the signaling via Pi3 kinase pathway. We'll discuss it in our future slides, and know how the control is done, and how a single mutations of the protein, simple structural change of the protein can leads to the malfunctioning of the signaling pathway, and what is the effect of this malfunctioning signaling pathway in cellular integrity. Okay.